Hello, my name is Donovan Ashby. I'm a postdoctoral fellow in the Hotchkiss Brain Institute at the University of Calgary. I'll be talking today about a project combined mesoscale cortical imaging and subcortical electrophysiology in awake mice. This project is supported by the Center for Open Neuroscience platform. The aim of this project is to develop a hardware-agnostic approach to simultaneously recording subcortical brain activity using electrophysiology in combination with mesoscale cortical imaging. First, we'll talk a little bit about what mesoscale cortical imaging is, talk about the rationale behind integrating subcortical electrophysiology to examine cortical-subcortical interactions. We'll run through the technical approach with regards to the hardware, the technical approach with regards to the software, and look at an example project where we're recording hippocampal spiking during resting state cortical activity. We'll look at one example of how to analyze this data using spike-triggered maps, and then run through some of the extensibility and the limitations of this approach. My hope is that this overview will give you an idea of the utility of this approach and its limitations. Cortical imaging. It's using wide field imaging to measure activity at the dorsal neocortex surface of the mouse. The dorsal surface of the mouse is optically accessible, and using modern super bright fluorescent biosensors, there's no requirement for skull thinning or removal. As you can see, a wide swath of the mouse cortex is optically accessible, and while this method does not allow resolving individual neuronal transients, as you will see, it provides a highly detailed measure of regional calcium fluctuations in multiple regions of the cortex. The base preparation involves affixing a glass cover slip to the skull, which provides an approximately 8 by 8 millimeter imaging window. Additionally, stainless steel hex bolt is affixed posterior to the imaging window to allow head fixation. In this case, we're using the Thai-1 JR gecko transgenic mouse, which expresses a redshifted calcium indicator, which has some favorable properties for wide field imaging applications. After habituation to head fixation, resting state cortical activity is imaged with an overhead camera. In the case of JR gecko, this involves excitation with a 567 nanometer LED and emission filters. This is an example of resting state cortical activity played at 1/4 speed. On the left are raw pixel values, and on the right are those pixel values expressed as a delta F over F. Clearly, there are structured dynamics in resting state cortical activity, and one simple question is, how is the activity in major afferent and efferent subcortical regions related to these ongoing cortical dynamics? Integrating subcortical electrophysiology. Cortical dynamics are organized in part by cortico-subcortical interactions, and understanding how cortex-wide neural activity relates to activity in these subcortical targets is necessary to understand ordered and disordered cortical processing. So as an example, we will target hippocampal-cortical interactions. Hippocampal-cortical information flow is crucial for mnemonic processing, among other functions. So we will adjust our base preparation with a small craniotomy positioned posterior to the imaging window. This craniotomy allows the insertion of a single or multi-site electrode, and we can precisely control the orientation and position of this electrode with a multi-axis micro manipulator. This approach is meant to be hardware agnostic, but in this case we're using Spike Gadget's electrophysiology recording system along with their excellent open source acquisition system TRODES in combination with a commercial mesoscale cortical imaging system by Labeo Technologies. Since imaging frame rates are several orders of magnitude slower than ePhys acquisition rates, the simplest way to synchronize across modalities is to record your frame triggers on your ePhys acquisition system. So presuming you have both modal systems independently operational, the only thing you need is a single BNC splitter. software technical approach. There are several modality-specific pre-processing steps that we'll only touch on briefly here. For imaging, this typically involves hemodynamic correction, movement correction, possibly temporal filtering, expressing values as a delta F over F, and possibly ROI extraction. For electrophysiology, this would typically involve digital referencing, filtering, and spike sorting. Once you have completed modality-specific pre-processing, you should be left with two data sets, a processed image stack and a set of electrophysiological recordings. At minimum, you will have a digital in time series containing your frame triggers, along with at least one electrophysiological signal. These could be local field potential fluctuations or spike time series. The available software utilities extract frame triggers from the digital signal and can generate two types of image-based analyses. The first is spike-triggered maps, wherein a stack of images is generated for each spike from a given cell. And the second is a seed signal correlation map, where each pixel is taken as a time-varying signal and is correlated against an electrophysiological signal to generate a correlation map. ROI-based analyses are also available, which are again based on cross-modal signal correlations. Let's take a look at an example recording of a single hippocampal neuron recorded alongside cortical resting state activity. At the bottom you can see action potentials recorded from a single hippocampal neuron. As you can see, the spiking activity is associated with ongoing cortical activity. Let's see how activity in this neuron looks when analyzed using spike-triggered maps. At top we can see a montage of activity and the associated spike-triggered maps. By inspection we can see that spikes are often occurring concurrently with retrosplenial cortex activity. At the bottom here we can see the average map color-coded, as well as the location of the peak calcium response for each spike in each hemisphere from the same cell as above. We can clearly see that retrosplenial cortex activity is dominantly associated with spiking in this particular cell. When we compare the average spike-triggered maps across neurons and plot the location of the peak calcium activity from each cell in each hemisphere, we can clearly see that spiking activity in hippocampus CA1 is predominantly associated with retrosplenial cortex activity. Thus, the principal features of hippocampal-cortical interactions during resting state can begin to be described using this very straightforward spike-triggered map approach.
Conclusions. Multimodal integration can be implemented to examine brain activity and to address long-standing questions, particularly related to cortical-subcortical interactions. Generic acquisition systems can be used, mostly because they have TTL I/O capacity, which is a very simple and very flexible tool that allows for synchronization. There's no need for bespoke tools, and really the key to this is open source software. As I hope you can imagine, the methodology presented here can be adapted to different subcortical targets and different preparations. There are some limitations, the first of which is that mesoscale cortical imaging for the most part requires head fixation. While head fixation is well tolerated, it limits behavioral assessments.
There are several notable limitations. The first is that mesoscale's cortical imaging in general requires head fixation. While it's well tolerated, it limits the scope of behavioral assessments that are possible. A natural extension of this would be to virtual environment exploration. As well, the subcortical targets that could be accessible with electrophysiology are limited, notably those regions that are centrally located just below the cortex, such as anterior thalamic and posterior striatal regions may be inaccessible. Thank you very much. In particular, I would like to thank the Center for Open Neuroscience Platform, as well as Labeo Technologies, the Hotchkiss Brain Institute, and the McGurr Lab.